Students, continue our previous class regarding meiosis. As we have already discussed meiosis till stage diakinesis. We have seen in prophase first, and that was completed in lipotine, in zygotine, in pecatine, in diplotine, and we have also seen what is diakinesis. So till to this stage in meiosis first division we have reached up to diakinesis. Now we may today in this second part of this class, meiosis first class, we may discuss metaphase first, anaphase first, and telophase first, and in this way our meiosis first will get completed. So what happened in metaphase first? The bivalent chromosomes which are there, which you have seen during diplotine, that there occurs synaptomial complex structures or bivalents. They now come to the equatorial plate, that is, they try to align in the center, or we may say they try to arrange themselves on the equatorial plate at the central line. So in metaphase first, what happened? The bivalent chromosomes, they start aligning, they start arranging themselves, they start placing themselves on the equatorial plate, and the microtubules from the opposite poles of the spindle attach to the pair of homologous chromosomes. That is, this is the figurical diagram which we are trying to show you, and this is the metaphase first. These are the bivalent chromosomes which tend to arrange themselves at the equatorial plate, and this is microtubule in north pole of the cell, and this is microtubule in the south pole of the cell, and they are here attached with these homologous chromosomes through spindle fibers. So this is all what happened during metaphase first in meiosis first, and now we may see what happened in anaphase first. So this is the third phase, that is anaphase. First one is prophase, second one is meta, third is anaphase, and last one is telophase. And we have seen the prophase first, that is divided into subphases, and those were zygotine, pecatine, diplotine, and finally diakinesis. But here these phases, they are simple and they are short. So when we are in anaphase first, here what happened, the homologous chromosomes which are here aligned at the equatorial plate in metaphase first, they separate now. They separate from each other. They tend to move towards the opposite poles. Some move to upwards and some move to downwards. Some move towards North Pole and some move towards South Pole, and they remain attached with the microtubules through these spindle fibers. But while this separation, the sister chromatids, they remain associated at their centromeres. They don't split, they don't separate sister chromatids, but they remain in contact. They remain in contact with their centromeres. So this is the pictograph, this is the diagrammatic structure what happened during the anaphase first division of meiosis first. And simply in this anaphase first, the homologous chromosomes, bivalents, which are so arranged at the equatorial plate in metaphase first, they now tend to separate from each other, but their sister chromatids, they remain intact, they remain attached, they remain in association with their centromeres. And finally, we may see here what happened in the last phase of meiosis first, that is telophase first. This is the last stage of meiosis first. And meiosis first, as you also know, it is known as heterotropic or reductional division because the chromosome number is reduced to its half. So what happened in telophase first? The nuclear membrane and nucleolus reappear. They the nuclear membrane and nucleolus, they start their appearance again. They are now trying to show their appearance again. They reappear again, and cytokinesis follows, and this is called as dyad of cells. Cytokinesis means division of cytoplasm. It follows here, and this stage where cytokinesis is followed, this is now known as dyad of cells. This all happen in telophase first. The stage between the two meiotic divisions is called interkinesis. So the stage between two meiotic divisions, that is meiosis first and meiosis second, this time interval or this period is known as interkinesis, and this interkinesis, it is very short period. And interkinesis is followed by prophase second. Now this enters, after entering into the interkinesis, this cell, it now again enters into the meiosis second division, and the first stage of meiosis second division is prophase second. So tomorrow our next class will be regarding meiosis second, and there we will see what is prophase second, what is anaphase second, metaphase second, anaphase second, and telophase second, and what happened there. And finally we will see that what is the importance of meiosis or meiotic divisions. So here the nuclear membranes, they occur, and this cell, it enters into interkinesis, and finally after interkinesis they are now followed by prophase second. So in this way we have we have completed meiosis first division, and we have discussed it in details that what happened in phase wise, in stage wise, to the cell during the meiosis first division. And inshallah in our next class we will see what is meiosis second. Till then, goodbye, have a good day, thank you.